Assalamualaikum and good morning. My name is Tihani Sofea Binti Abdul Latif, and today I would like to present with you my research proposal, which is the differences in amino acid metabolite production in Chlorella species during UVB exposure. Before we go any further, let me briefly explain about Chlorella. Chlorella is a unicellular spherical green microalgae that contains chlorophyll pigment and is widely studied for its high content of essential metabolite. When exposed to environmental stresses, microalgae will activate their defense mechanism, leading to the production of specific metabolite that help protect against potential toxicity. As we all know, global warming and deterioration of the ozone layer are pressing environmental issues. These changes in our environment increase the level of UV radiation reaching the Earth's surface. One of the primary concerns is the impact of UVB radiation on photosynthetic microorganisms like Chlorella. This is because prolonged exposure to UVB can lead to cell damage and alter the metabolic activities within these cells. The research question of this study include when is the stationary phase of Chlorella culture, what is metabolite produced in stationary phase of Chlorella culture in response to the UVB radiation, and is there a difference in metabolite level that are produced in UV exposed and non-UV exposed of Chlorella cell. Therefore, the aim for this research is to determine the stationary phase of Chlorella culture as well as to identify the metabolite produced in the stationary phase in response to the UVB exposure. The production of metabolite level in UV condition and non-UV condition will also be compared throughout the study. Understanding the impact of UVB exposure on Chlorella will shed light on the microbiome resilient to UV damage. Metabolome analysis will provide significant knowledge regarding the interaction that occur between the organism and its environment. Understanding how these microbes modify their metabolism is essential for evaluating their survival in changing environment. First of all, let's take a look at the effect of UV radiation on microalgae cells. UV radiation can harm microalgae growth and metabolism by damaging crucial biomolecules. DNA can get damaged by disrupting transcription and replication, leading to mutation and higher cell mortality. UVB radiation also triggered the production of reactive oxygen species, which further damaged the cell. A 2019 study found that 98.2% of Chlorella cells had intact membrane before UV exposure. However, the damage rate increased significantly up to 40.1%, with cell losing integrity and even lysing. Therefore, to cope with UV radiation, microalgae have developed several defense mechanisms. First, they migrate to deeper water to avoid excessive UV exposure. Microalgae also activate non-photochemical quenching, which release excess light as heat with the help of orange carotenoid protein. To remove the excessive ROS, microalgae use antioxidant enzymes like superoxide dismutase and catalase. Lastly, they produce UV-absorbing compounds like microsporin-like amino acid and histamine, since these compounds have a photoprotective activity. Amino acids are essential metabolites in Chlorella, which are categorized into families like glutamate, serine, shikimate, pyruvate, and aspartate based on their metabolic intermediate. Metabolites are generally divided into primary and secondary type. During the log phase of Chlorella's growth, the cells show peak metabolic activity, focusing on producing primary metabolite. As they enter the stationary phase, the production shifts towards secondary metabolite. Any disruption in amino acid synthesis during the primary phase can limit precursor for secondary metabolite, which affect the cell's ability to adapt and survive in changing environment. UCMS is the gold standard in metabolomics due to its high sensitivity and specificity. It separates volatile molecules in gas chromatography and then ionize and analyze them by mass to charge ratio in the mass spectrometer. GCMS is able to run and analyze many compounds, which is around 400 compounds, in a single run. Now, let me outline the methodology involved in this study. The Chlorella organism will be cultivated in BG11 liquid medium at 24 degrees Celsius. The culture is continuously exposed to photosynthetically active radiation to support their growth. To determine the growth curve, two methods are used. The first is the indirect turbidity method, where samples are taken at regular interval, which is 0, 24, 48, and 72 hours, and the turbidity is measured using spectrophotometer at 750 nanometer. The optical density for each interval is recorded and plotted against time to generate the growth curve. The second method is direct microscopic cell counting using a hemocytometer. A small sample is placed on the chamber and a cell are counted under microscope. The cell density is calculated and the data is plotted to generate the growth curve, which help identify the stationary phase for UVB exposure. For the irradiation of Chlorella, culture from the stationary phase are transferred into therapeutic dish and exposed to the UVB radiation using UVB lamp. Samples are collected every 24 hours to monitor changes over time. For comparison, another set of culture is kept under normal condition without UVB exposure and also collected at the same interval. After irradiation, both UV and non-UV samples are centrifuged to separate the pellet and supernatant. The supernatant is discarded and the pellet is freeze-dried. The pellet is then resuspended in methanol, water, and chloroform mixture and sodicated to break open the cell and release intracellular content. The sample is further concentrated by a rotary vacuum concentrator to remove remaining solvent. The metabolites are then derivatized by mixing with MSTFE and pyridine and heated at 80 degrees Celsius for 20 minutes. Once cooled, 0.5 microliter of the sample is injected into the column in a splitless mode. In this GCMS analysis, helium is used as the carrier gas. The metabolites are then identified using the NIST 2014 library database. Statistical analysis is performed based on the obtained data to compare the level of metabolite in UV condition and non-UV condition. The expected outcome for this research study will reveal the alteration of metabolite profile in Chlorella organism. Some metabolites are unique to non-UV condition, while others are specifically produced after UV exposure. There are certain metabolites that will be activated or downregulated in response to the UV exposure, leading to varying concentration depending on the environmental stress. However, challenges such as fluctuation in environmental conditions and contamination may arise during this study. Factors like temperature, humidity, and light intensity can affect metabolic processes and lead to inconsistent results. To mitigate this, experiments should be conducted in controlled environment with standardized conditions. Contamination from microbes can also alter metabolite production, hence sterile handling and equipment is significant. I think that's all from me. Thank you for your attention.